Good morning. My name is Jonathan Hicks, and I want to welcome you to the NIH Office of Disease Prevention Medicine Mind the Gap webinar series. This webinar series explores research design, measurement, intervention, data analysis, and other methods of interest to prevention science. Our goal is to encourage the prevention research community in thought-provoking discussions to promote the use of the best available methods in prevention research and to support the development of better methods. Before we begin, I have some housekeeping items. To submit your questions during the webinar, there are two options. First, you may submit questions via the WebEx by clicking on the question mark in the WebEx toolbar and direct questions to all panelists. Second, you may participate by Twitter and submit questions using the hashtag NIHMTG. At the conclusion of today's talk, we will open the floor to questions that have been submitted via WebEx and Twitter. And lastly, we appreciate your feedback about today's webinar. Upon closing the webinar meeting, you will be directed to a website to complete a seminar evaluation and also send an email with a link to the evaluation. Your insights will help us to continue to improve this webinar series. At this time, I'd like to turn things over to Dr. David M. Murray, Associate Director for Prevention and Director of the Office of Disease Prevention.
Thank you, Jonathan. I'm going to introduce Dr. Jason Moore, who's the Edward Rose Professor of Informatics and Director of the Penn Institute for Biomedical Informatics. He also serves as Senior Associate Dean for Informatics and Chief of the Division of Informatics in the Department of Biostatistics, Epidemiology, and Informatics. He came to Penn in 2015 from Dartmouth, where he was Director of the Institute for Quantitative Biomedical Sciences. Prior to Dartmouth, he served as Director of the Advanced Computing Center for Research and Education at Vanderbilt University, where he launched the first high-performance computer at that institution. He has a PhD in human genetics and an MS in applied statistics from the University of Michigan. He leads an active NIH-funded research program focused on the development of artificial intelligence and machine learning algorithms for the analysis of complex biomedical data. He's an elected fellow of the American Association for the Advancement of Science, an elected fellow of the American College of Medical Informatics, an elected fellow of the American Statistical Association, and he was selected as a Cobley Fellow of the National Academy of Sciences. He is currently a Penn Fellow and serves as Editor-in-Chief of the journal Biodata Mining. It's my pleasure to welcome Dr. Jason Moore, and I'll turn the session over to him.
Great. Good morning, and it's great to be with all of you online. Thank you for joining. What I want to accomplish today is to introduce a hot new area called automated machine learning. This field is only a few years old, and it's rapidly evolving, but I think it has the potential to have a big impact in biomedical research and certainly prevention research. But before I start, I just want to say if you see something that interests you, if there's a way we can help you with your research, please don't hesitate to contact me, or if there's a way we can help you use some of our automated machine learning software, please reach out to me. You have my email address, and we'll do our best to help you. So I'm a biomedical informatician, and I thought it would be good to start the presentation from that point of view. And if you're not familiar with the discipline of biomedical informatics, this is how the American Medical Informatics Association breaks down the different components of this broad discipline. So they break it down into a basic science component, what we think of as bioinformatics and computational biology, really focused on more basic science questions using omics data. The clinical informatics piece is split into three sections. Clinical informatics, which is really about designing computer systems and databases to deliver healthcare, things like clinical decision support used by clinicians in the healthcare environment. Clinical research informatics, which is an exploding area using electronic health record data, other clinical data to answer research questions that might have an impact on healthcare. And then consumer health informatics using smartphone technology, for example, to monitor patients in their home environment, or telemedicine to allow them to communicate with their healthcare providers remotely. And then, of course, public health informatics using informatics methods to impact the health of populations. I published an editorial in Biodata Mining last year with John Holmes, making the argument that this is the beginning of the golden era of biomedical informatics. This is a very exciting time to be an informatician because we're swimming in big data. We now live in the big data era. We're all dealing with or attempting to figure out how to deal with big data. High performance computing is inexpensive and plentiful and accessible. We now have a number of years under our belt of training informaticians, data scientists, et cetera, and those individuals are now joining faculty, joining the job market, and having an impact. So we have a number of talented trainees around now. Certainly, as many of you are familiar with, we have government recognition of the importance of informatics and data science. The NIH has certainly embraced this across the different institutes. We have tremendous industry recognition. In fact, industry is hiring informaticians and data scientists as fast as we can train them and competing with us directly for those individuals. As an example, we did a faculty search last year here at Penn between the Department of Genetics and the Institute for Biomedical Informatics to recruit informaticians working in the genetics and genomics bioinformatics space. Our top 2 candidates ended up going to Google instead of taking academic positions, and they could have gone to any institution in the country. There's certainly patient recognition. Patients now have access to their own healthcare data. Electronic health records like Epic have web portals now where you can log in and see your test results, communicate with your physician, order prescriptions, et cetera. And university investment has never been bigger. At a time when a lot of universities and medical centers, medical schools are contracting due to reduced federal funding and other budget constraints, state funding constraints, universities are choosing to invest heavily in data science and biomedical informatics, starting new departments, new centers, and again, hiring faculty as quickly as they can get them. So if this is the golden era, and I think it is, and what can we expect over the next 10, 15, 20 years? I think artificial intelligence is going to play a prominent role, and we've seen a number of important advances in AI over the last few years. Biomedical devices are exploding and having an impact in healthcare and generate massive amounts of data. Data integration, we collect lots of research data, clinical data, but putting them together to answer questions is important. This is a really hot area. Data science, of course, integrating mathematics, computer science, and statistics to answer questions. The idea of informatician scientists is an idea that I've been pushing, that informaticians are in a perfect position to have an impact on biomedical research because we can look across data sources, information sources, knowledge sources, and integrate all of that information to ask more impactful scientific questions. I'm going to talk today about machine learning, which is becoming a very, very hot and accessible area. No boundary thinking is again this idea that informaticians can look across knowledge sources to across different disciplines to ask more impactful questions that might be missed by people only familiar with their specific literature. And visual analytics, which if there's time, I'll say a few words about it at the end. So this is how I think about common complex diseases like cancer, cardiovascular disease, neuropsychiatric diseases, and I've illustrated here bladder cancer, a disease that I've worked on for at least 10 years. So at the bottom of this slide, we have the human genome and its 20,000 or so genes and non-coding regions, and those genes code for proteins and enzymes that are part of numerous biochemical systems, physiologic systems, and of course all this complex biology happens in the context of our local ecology, our environmental exposures, our diet, exercise, smoking, etc. I have illustrated here arsenic, which is a toxic metal that's believed to be a risk factor for bladder cancer that we ingest in our drinking water and our food that sits in our bladder and potentially damages our cells, our DNA. And it's all of these things working together in a complex way that determine our individual trajectory shown at the top here through time toward a complex clinical endpoint such as bladder cancer. And what have we done in human genetics and epidemiology? We take single factors out of this complex picture, like a single nucleotide polymorphism, a single nucleotide variant out of the human genome, or a single environmental factor out of our ecology, and ask whether that single factor is predictive of risk of disease. This seems very unrealistic to me, given the complexity that we know exists that maps the human genome, the exposome, to our risk of disease. And so this is the real motivation for my research in machine learning and artificial intelligence is how can we develop the methodologies, the analytic methodologies that allow us to embrace this complexity rather than ignore it, to move away from the one factor at a time approach to looking at interactions, complex combinations of genetic and environmental factors that are predictive of disease. So this is the motivating factor for my research. Now, what I've illustrated here is my version of the data science pipeline. These are the steps that you would need to go through to go from data experiment to clinical application, for example. So shown on the left side is diverse data sources that we collect about patients. The next step is data integration, and I'll step through these one at a time here in a second. The next step is feature selection, feature construction, machine learning, interpretation, experimental validation, and finally clinical application. So I'll just step through these quickly. So of course we now live in the big data world. I like this slide. This is how I think about big data, that big data is not necessarily just one type of data, a lot of one type of data, but increasingly we're combining, integrating lots of diverse data collected from diverse sources, from diverse technologies, each with their own quality control issues, et cetera. So not only is data big and highly variable and coming at us very quickly, but it's very complex, and I think that's one of the pieces that's often overlooked when people talk about big data. Data integration, as I've mentioned, is very important. We often put data into relational databases, but a new informatics technology that's coming on the scene very quickly is what's called graph databases. I'm not going to talk about this anymore, but highly recommend those of you not familiar with it to look it up and get a little bit of exposure to graph databases. I think we're going to see this technology a lot more. But the basic idea is to be able to put diverse data into a database very quickly, and each of the individual data elements are linked to each other through keywords or metadata that then allows you to do a very complex semantic query across that data. So graph databases are highly flexible, allow you to get up and going very quickly, and allow you to link together very diverse data in a way that allows you to query it later. Now, big data means collecting a lot of variables, what we call features in computer science. And certainly in genetics, we can now measure millions of individual genetic variants, features in population-based studies. And the exposome technology allowing us to measure environmental exposures is quickly becoming very big. So what do you do if you're faced with 1 million or more features? It's very difficult to put all that into a single machine learning method. So often what we need to do is what's called feature selection, reduce that to a more manageable subset, but do it in a way that's informative. So shown at the top here are computational statistical methods for reducing the feature space. ReliefF is a method that we've worked on, which embraces the complexity of the problem and allows you to filter out the noisy variables from things that are more likely to contain signal. And shown at the bottom is biological feature selection, so selecting a subset of features based on your biological knowledge of the problem. For example, if you're studying diabetes, you might choose to focus on genes that are involved in insulin metabolism or specific dietary factors that are known to promote diabetes, for example. So using your biological knowledge to reduce the feature space. Another important but often overlooked area is what's called feature construction or feature engineering, and this is the idea that the data doesn't always come to you in the right format, that you might need to remap or represent the data differently to make it easier for the machine learning method to detect it. So on the left, I've shown two features. These could be two environmental factors or two genetic factors with discrete levels 0, 1, and 2. And then there are different ways that you can map those features into a new feature shown as the letter Z here. And it's that new feature that you then put back in the data set and include for analysis. And this is important because it can alleviate the machine learning method from having to learn that particular pattern. You've done some of the work for the machine learning algorithm for it. And then, of course, once you've done all this pre-processing, then you need to pick one or more machine learning methods to use for your data. And I like this slide a lot. If you haven't seen it before, I think it's a very informative slide that really illustrates what machine learning is doing. So shown on the left are some 3 different data sets. And the X and Y axes think of as 2 different continuous variables. So you might have age and weight, for example, and maybe the red dots are people with disease and the blue dots are healthy controls. And what you want to do is find a mathematical function that discriminates the healthy people from the sick people based on these two biomarkers or these 2 features that you've measured. And at face value, these data sets don't look too different, but then shown across to the side are 3 different machine learning methods, a nearest neighbor classifier, a linear support vector machine, and a radial basis function support vector machine. And then you can see in the color shading the probability, the darker the shade, the higher the probability that an individual is a case or a control based on those 2 biomarkers. And the point of this slide is to say that each method looks at the data very differently and maps those 2 biomarkers using different mathematical functions. And this is a key element of machine learning, is that each method looks at the data differently, and it's hard to know beforehand which is the right method to use. And you can see in the numbers in the boxes, those are think of those as the quality measure of how well the model fits the data. So the higher the number, the better. And you can see that different methods are optimal for different data sets. And so how do you know beforehand which to pick? How do you know which one's right for your data? This is a big question, and it means an important question, and it means you need to be familiar with a whole bunch of different methods, and maybe not rely on any one method, but try different methods. And then even within a method, there are different parameter settings that you might need to explore. And there can be a wide range of parameter settings for each of these methods, and knowing what parameters to select is an important challenge. And then once you've developed a machine learning model, I think the hardest part is then interpreting that. What does it mean? We've put a lot of work over the years into providing statistical and biological interpretation of the features, their relationships, what's really going on in a machine learning model, trying to remove some of the black box features. And I'll touch on this a little bit more later, because I think it's a very important step in all of this, and perhaps the most difficult. Then once you have something that's important, then you need to design an experiment to validate it, and hopefully you find something that will ultimately have a clinical application. So hopefully I've convinced you that this process of establishing a data science pipeline with machine learning is a very difficult process, can be very time consuming. It's very daunting even for machine learning experts. Those of us that work in machine learning day in and day out, struggle with how to build these pipelines and how to make all the decisions. It's not easy even for the experts. And so we asked the question, could some of this be automated to take the guesswork out and to make this technology more accessible to the naive user? And so we decided to focus on the middle part of this pipeline, doing feature selection, feature construction, and machine learning, and developing a computer algorithm that can automate all of that for you. So about 2 years ago, we started a project. This was led by a postdoc in my group, Randy Olson, and we developed what we call the tree-based pipeline optimization tool, or Teapot, for automating the middle part of that pipeline. The code for this is available on GitHub. It's all open source. It's all coded in the Python programming language, and we've got a number of people now around the world that are making contributions to the code base. So what I'll do is just step you kind of at a high level through the basic components of Teapot and to explain how it works and give you a little bit of a peek under the hood at what it's doing. So here are the 4 components that we needed when we developed Teapot. We needed a machine learning code base. We needed a way to represent pipelines in the computer so the computer could tinker with the pipelines. We needed a way to optimize for the computer to pick the best pipeline for a particular data set. And then, of course, we need to control for what's called overfitting. That is, we needed to make sure that all of this computational horsepower going into building a pipeline wouldn't just learn the noise in the data, that it would be able to gravitate to the signal. So that's what we call overfitting. So I'll just step through these one at a time. The first is the code base, and fortunately there is a very comprehensive, professionally run project to develop open source software for machine learning. This is all programmed in Python. This is the scikit-learn library from scikit-learn.org. And this is a very comprehensive collection of machine learning algorithms for doing classification, where you have a discrete endpoint, regression, where you have a continuous endpoint, things like cluster analysis, where you're not sure what the endpoint is, you're trying to discover the underlying structure in the data, dimensionality reduction, model selection, and a whole bunch of pre-processing algorithms that allow you to do things like feature selection that I mentioned, or normalize your data, or transform your data. So we take all the components of the scikit-learn library and use these as building blocks for pipelines. So this was great because we didn't have to recreate the wheel. We were able to use these open source tools. So here's an example of the building blocks from scikit-learn that we use. We have feature selection methods. You could, for example, do univariate tests on all your features and select those that look the most interesting, or do importance score sampling of your features. I'll show you an example of that in a second. You might do feature processing, things like normalizing your data, doing a polynomial transformation, scaling your data and coding it in some way. Or feature construction, principal components analysis, for example. PCA takes sets of features, puts them together in a linear combination, and finds the subsets of linear combinations that are statistically independent of one another, and then outputs that as new features that you can then use for machine learning. And then, of course, you have your machine learning algorithms, and like I said, there are a large number of machine learning algorithms in the scikit-learn library. I've just shown a few of them here, things like support vector machines, decision trees, random forests, and we typically gravitate for the more commonly used machine learning methods when we construct our pipelines. So we use these as building blocks, and here's an example of a hypothetical pipeline that could be constructed by Teapot. So you start with your data, and the data is fed down 2 different paths here. So on the top path, it first goes to a random forest algorithm that takes the features in the data set, builds a model to predict some endpoint, and then ranks the features based on how important they are. And you can see in the green box, it actually does feature selection. It selects a subset of features based on those importance scores, takes that subset, does a polynomial transformation of the features, feeds that to k-nearest neighbors machine learning algorithm, and then takes the output of k-nearest neighbors and feeds that to a decision tree. And then on the bottom path, it does principal component analysis, feeds those new constructed features to a support vector machine, takes the output of the support vector machine, combines that with the output of the k-nearest neighbors algorithm in a decision tree, and it's the decision tree algorithm that does the final classification or prediction on your data. Now, this is a fairly complex pipeline. There's a lot going on here, but it might be that there are several different machine learning algorithms, different subsets of features, different ways of representing the features that are important to really capture the underlying complexity that you're looking for in your data. So the question is, how can we find these pipelines and evaluate them, make sure they're not overfitting, but also make sure they're doing a good job of predicting. So we need a way to represent the pipeline in the computer. So on the top here is the pipeline I just showed you. On the bottom is how we represent it in the computer. We represent them as expression trees. This is a type of data structure in computer science that allows the computer to represent and manipulate the pipelines. So once we have, so the way this algorithm works is we start by generating a bunch of random pipelines, maybe 1,000 random pipelines. And then what we do is pick the best ones based on how good of a job they do at predicting your endpoint. We also pick them based on trying to minimize the complexity of the pipelines, and I'll show you a little bit more about that in a second. And then what we do is we want to pick the best pipelines, and we want to introduce variability. We want to try out new pipelines, so we want to tinker with these pipelines. So what we do is we treat the good pipelines from the population of all pipelines as parents, and then we want to change them. We want to exchange subcomponents of pipelines. We want to mutate or change individual components of the pipeline and generate children, new pipelines that we can then move forward and evaluate. So in this example, the pieces of the pipeline at the top in the oval are swapped. They're exchanged or recombined to create two new pipelines. So these are, and this makes sense, right? Because you can imagine that subcomponents of different pipelines might be useful in and of themselves, and putting them together with other subcomponents might generate much better pipelines. So that's what's shown in this example. We're swapping subcomponents of pipelines, and then what's shown in with the star, the yellow star, is that we are changing an individual component. So we're changing the polynomial feature transformation piece of that pipeline to one that looks at variance. So in this way, we generate variability in the pipelines, and then we can look at the new pipelines and go through this iterative process of, again, selecting the best pipelines, changing them, and iterating until we meet some criteria, either some stopping criteria or some performance criteria, and then we stop the algorithm and take the best pipelines to look at. So if any of you in the audience are statisticians, you're probably squirming in your seat right now because you can imagine the overfitting issue that might arise with piecing together different machine learning algorithms like this. That's certainly something that we worry about and are concerned with, that the pipelines might be really good at learning the noise in the data and not focusing on the signal. So we do two things to try to combat that. The first thing we do is cross-validation. The value of a machine learning method is not its ability to describe a data set, but its ability to generalize an independent data. A lot of interest in replicability and reproducibility of results, and so this cross-validation is a way to try to address that. So here we take the data, we split it into two pieces, the 75-25. We take the 75 piece, split it into two additional pieces, use the one piece, the 75%, for training. So we run the algorithm on that piece of the data. And then when we pick what we think are the final best pipelines, we can then apply them to the testing data, the 25%. And then pick an overall best pipeline and then apply it to the validation set, which is a piece of data that the algorithm hasn't seen yet, to really assess the generalizability. Does this algorithm generalize to independent data? if it's learned the noise and the training data and the testing data, it should be specific to those pieces of the data and not generalized to the validation data. So that's one way we control overfitting. The other way we address this is through what's called Pareto optimization. So on the y-axis here, we have the error of the machine learning model. So this is the traditional way to measure the quality of a machine learning model, is how good of a job is it doing at predicting your particular endpoint. But we add a second piece of information here, which is the complexity of the pipeline on the x-axis. So here, we're also looking at the complexity, which could be measured by the number of individual algorithms that are being used in the pipeline, the size of the pipeline. And there's a trade-off here that is captured in this Pareto optimization. So shown in the white dots, if you look at the top left, is a model that has very low complexity, so that might be of interest because it's a simpler pipeline, but the error is not very good. And if you look at the white dot on the bottom right of the plot, here you have a model that has a very low error. So statistically, it's a very good predictor of your endpoint, but it's also very complex and might have many different machine learning models in it. And in that sense, the complexity might not be of interest to you because it'll be difficult to interpret or understand what the pipeline is doing. And so what Pareto optimization allows you to do is explore all the white dots, and this is how we select pipelines for the tinkering process that I mentioned previously. And the sweet spot might be in the middle there, where you balance error and complexity and have a trade-off here. You're willing to sacrifice a little bit of error for a simpler model, and that might be the pipeline that you ultimately decide to look at. So these two things in combination, the cross-validation and the Pareto optimization in our experience, do a pretty good job of combating the overfitting problem. They're not perfect, but I think they do a good job, and that's certainly been true in some of the simulation experiments that I'll show you. Okay, so let me show you the first thing we did when we developed Teapot is we wanted to do some computer simulations where we knew what the answer was and could really evaluate whether Teapot is finding the answer and how often it's finding the answer and how does it compare to a traditional machine learning approach. So here are the results of a simulation we did. This was in a computer science conference proceedings paper that we published last year, which actually won a Best Paper Award at the conference where we presented this. Shown across the top of these different panels are the sample size going from 200 to 1,600. Think of those as human subjects in your data set. And what we're doing here is simulating genetic data. And then, so along the y-axis of these plots is the heritability of the models that we use to simulate the data, so essentially the effect size of the genetic biomarkers. So the top is a stronger genetic effect size, and at the bottom is a weaker genetic effect size. So you can imagine at the bottom left plot, the most bottom left plot is the smallest sample size and the weakest signal. So that's where the algorithm should perform the worst. And then in the top right, you have the biggest sample size and the biggest effect size. So this is where the signal is the strongest and the algorithm should perform the best. And that's what we see. And then on the y-axis of each plot is the accuracy of the classifier. So a higher value is better. That means it's classifying more human subjects correctly. 0.5 is the baseline, what you get if you just flip a coin and to predict, say, case control status in a population-based study. So the further away the box plots are from 50%, the better they are. So the box plots represent 30 independent runs of the different algorithms, and then the white box is just a plain vanilla random forest algorithm. So that's what you get if you just run a commonly used machine learning algorithm on the data. And then Teapot is in the with a search algorithm is the rightmost two box plots. And so you can see that as the sample size and the signal increases, Teapot is doing better and better. compared to a baseline random forest algorithm or Teapot with just models and not all the other pre-processing stuff. So this convinced us, the simulation convinced us that Teapot was working, that it was finding a complex signal in the data set, that the pipelines that Teapot was building were better than just running a standard machine learning algorithm as most people would right out of the box on these data sets. So this was a nice result and allowed us to confirm that this new method we had developed was indeed working. So what I'd like to do now is show you a real data example with Teapot. This was the first real data set that we applied Teapot to, and I think it's a nice example because it's a small data set. We have previously published this data. The signal in this data was replicated in an independent data set, so we know that the signal replicates. So there was something here to find. It's a complex signal. So this is a bladder cancer case control study from the state of New Hampshire. These are Caucasian subjects, so a relatively homogeneous population. This was a study that was designed by Margaret Carragus at Dartmouth College and her colleague Angeline Andrew, two cancer epidemiologists. And initially they were interested in DNA repair enzyme genes as candidates for predicting bladder cancer, thinking that bladder cancer is partly due to toxic metals and other factors, things in cigarette smoke, for example, that damage DNA and increase risk of cancer. So DNA repair enzyme genes are thought to play an important role in protecting against DNA damage and cancer. So they looked at polymorphisms in a couple different DNA repair genes. They also had peck years of smoking, age, and gender in this data set. So we had previously analyzed this data set using a machine learning method that we developed called multi-factor dimensionality reduction, which is designed specifically to detect non-additive interactions between biomarkers and data sets. And this is a result we published in Carcinogenesis about 10 years ago. And this is the final best model that we found, doing machine learning by hand in this data set. So what you're looking at are 3 different biomarkers, 2 different polymorphisms in the xeroderma pigmentosa gene, the XPD gene, shown on the Y and X axis of these plots. The genotypes are coded 0, 1, 2. And then shown at the top, the different panels are pack years of smoking. So on the left side, you have subjects that smoke less, and on the right side, you have subjects that smoked a lot. The bars represent, the left bars are the number of cases, people with bladder cancer, and then the right bars are the number of people in the data set with the number of healthy controls without bladder cancer. The dark shaded cells are what the algorithm determined were high risk, and the light shaded are the combinations determined to be low risk. And you can see that on the right side of the panel, people that smoked more were at higher risk because you have more dark gray shading, and on the left side you have more light gray, which is lower risk, people that smoked less. You can see the main effect of smoking very clearly in this plot. But note the kind of crisscrossing pattern of high risk and low risk across the different genotype combinations within smoking. This is indicative of non-additive interactions, and I'll show you more of those details here in a second. And then what MDR does, this MDR algorithm, is it's actually a feature construction algorithm. So it's taking those 3 biomarkers and then mapping them into high risk and low risk combinations, and then including them, mapping them to a new feature shown at the bottom here with G0 and G1. So all the data is remapped into this single feature, and then it's that new single feature that MDR uses to make a prediction of high risk and low risk. So this model correctly classified bladder cancer in the training data 66% of the time, and the test data 64% of the time. If you look at that single 3 biomarker feature has an odds ratio of about 3 with a significant confidence interval. And then if you do a 1,000-fold permutation test, you randomize the case control labels 1,000 times, repeat this entire analysis, a model this good was never found in the randomized data. So in other words, it had an empirical p-value of less than 0.001. Okay, so this is work we've previously published and replicated. We kind of knew what the signal was in this data set. We've looked at it with a number of other machine learning methods. So what we wanted to do was apply Teapot to this data to see if Teapot could discover the signal here that we knew was there. So here are the building blocks that we included in Teapot. Feature selectors, feature processors, feature construction algorithms, and we included MDR in here. We didn't tell it to use MDR, but we included it as one of the options to see if Teapot would discover what we had previously shown, that MDR does a good job of picking out the signal in this data set. And then we included decision trees and naive Bayes classifiers as the machine learning algorithms. So we ran Teapot a number of times, and this was the best model that it found. And it actually did use the MDR feature constructor in the pipeline, and combined 2 rounds of MDR feature construction with a decision tree. So in the first MDR step, it's looking at the pairwise combinations of all the biomarkers here, and then shown in the light pink boxes, it puts the 2 XPD polymorphisms together in one constructed feature. So it's doing the same thing I showed you here. It's taking the 2 XPD polymorphisms and mapping that 2-dimensional space down to a single new feature shown in the light pink box. And then it also took pack years of smoking and age above and below 50 and constructed a new feature with those 2 biomarkers. Then those pairwise constructed features are then fed to another round of MDR, shown in the darker, bigger pink box here, and is taking those new constructed features and putting them together into a new feature, and then feeding that to a decision tree, which does the final classification. Now, the testing accuracy from cross-validation of this model in Teapot was 0.64, which matches exactly what we saw before with MDR. Now, this was reassuring because the Teapot pipeline is using MDR, using feature construction, combining the same genetic biomarkers. It's finding pack years of smoking. It's including age as an additional biomarker that was not found in our original MDR analysis. So this was very, very reassuring to us and matched very closely what we had found before, what we knew was the signal in this data set. Now, what I'm going to show you is the interpretation of this to show you exactly what this pipeline is doing. So that's shown on this slide. So here we show on the left side the individual biomarkers, the 2 genetic polymorphisms on the top, age and pack years of smoking on the bottom. And you can see that people that are older are at higher risk of bladder cancer. People that smoke more are at higher risk of bladder cancer. That's the dark shading there with pack years of 2 and age above 51. And then with the genetic biomarkers, it's less clear what the effect are. Actually, these 2 genetic factors have almost 0 effect on disease risk. So then what MDR is doing in this first step of the pipeline, shown here, is it's taking the 2 genetic factors, putting them together in 2-dimensional space, and doing the same for smoking and age, and then collapsing that 2-dimensional space to a single factor shown in the middle there, the single constructed feature. Okay, so that's the first part of this pipeline. And if you look at the 2 genetic features together, they combine, you can see the non-additive pattern of the genetic effects there, that kind of diagonal crisscross pattern. Okay, and the second part of this pipeline here it's taking these 2 constructed features and putting them together into the 2x2 box shown on the right side of the slide, and then doing a final feature construction to make a final feature, and that's what's being passed to the decision tree. And so this is a nice example of what I was saying earlier, that the feature construction piece of your pipeline can actually do the heavy lifting for your machine learning algorithm, and that's exactly what's happening here in this pipeline. The feature construction is doing all the hard work. Once you get that single constructed feature at the end here and feed that to a decision tree, the machine learning part of this is actually very simple. You could do univariate logistic regression at this point, and so it's a good reminder to put a lot of effort into the feature selection and feature construction part of the machine learning analysis. And actually, that's exactly what we were doing here in this particular study, only looking at DNA repair enzyme genes, is doing a biological feature selection that I mentioned earlier in the presentation. So there's a biological feature selection happening here, and then a feature construction happening with the help of this MDR algorithm before it ever gets to the machine learning step, the classification step. And then this is just an interpretation piece using information theory to look at the nature of the statistical effects. So this is the numbers here in this plot are, the higher the number, the more information is provided about case control status by an individual factor or a pairwise combination of factors. So you can see the main effect of smoking, pack years 3.65, so it has the biggest main effect. You can see the two XPD polymorphisms are almost zero. They provide almost no information individually. But when you combine them in the red line there, jointly they provide more information about bladder cancer than smoking does by itself. And that's a nice example of a nonlinear, non-additive, synergistic interaction between these two genetic factors. And if you were to look at this data one factor at a time, doing a univariate analysis, you would completely miss these two genetic factors as having an effect on bladder cancer. You would only pick up smoking and perhaps age as your significant factors. So this is why we do machine learning. Machine learning enables us to find these unexpected results, the results that would be missed in, say, a traditional parametric statistical analysis. Okay, so almost out of time. What we've been working on this past year is scaling Teapot for big data. We added an expert knowledge feature selector to allow you to load in a source of expert knowledge that Teapot can use to select a subset of features. It could be anything, could be prior statistical results, could be biological results. And this allows Teapot to look at much bigger data sets. I'm going to skip the simulation that we did, but we were able to show with as many as 5,000 genetic features that Teapot was able to pick out the right signal and do so better than traditional machine learning algorithms. So one of the questions I get a lot from people about when I present Teapot is you know, is this going to replace the data scientists? And I don't really see this as a replacement. I see Teapot and similar automated machine learning methods as more of a data science assistant, right? It's an extra set of hands in your research lab that can look at your data, generate a machine learning pipeline, and you can use those pipelines in different ways. You could use it directly. You could just say, okay, this is the best pipeline. This is what I'm going to use to analyze my data. This is what I'm going to report to the community. Or you could take the pipeline as a starting point and tinker with it and see if you can make it better. Or you could look at what Teapot's doing. What are these pipelines doing? If your Teapot pipelines are using random forest algorithms, then maybe that gives you a clue that random forest is the right machine learning algorithm to use on your data set. And you could then ignore the pipeline and just start with a random forest analysis and just use Teapot for ideas of a starting point. So I really see these methods as a data science or machine learning assistant rather than replacing the data scientist. And one of the things we're working on now is making all of this easy and accessible. I don't have time today, but we're working on a project right now to make this as easy as using an app on your smartphone. And if you're interested in automated machine learning, I run this informational website. I just put this up a few weeks ago at automl.info. where you can get more information about automated machine learning, some of the tools we're developing, and I've got listed in here some tools that other people are developing as well. And I'll be continually updating this website with new information. Like I said, this is a hot new field. It's really only been around for a few years and is evolving very quickly. And I'll just say that we're implementing this here at Penn, this and other similar tools in what we call the Idea Factory, which is Penn's first immersive visualization facility. And I think at the end of the day, these automated machine learning and AI tools are going to be much more useful when combined with state-of-the-art visualization and interactive computing through visual analytics. I think virtual reality, for example, is going to play an important role in exploring machine learning models. So I'll stop there and take questions. I just want to thank Randy Olson, postdoc now, senior data scientist in the lab, who did all the hard work on developing Teapot and wrote the code for Teapot and manages and continues to work on the project. Andrew Son is a medical student that's just is in the middle of a 2-year research term in my lab. Worked on the expert knowledge feature selector for Teapot. These are my NIH grants that have supported this work. And my contact information, I'm on Twitter @morejh. If you follow me, I'll do my best to follow you back. And like I said, feel free to email me with questions or ideas about how we can help you or collaborate. So I'll stop there for questions.
Thank you, Dr. Moore. Very interesting presentation. Thank you very much. And you had a very good-sized audience throughout the presentation. I'll start with a few questions. This is clearly a sophisticated and complex technology is one way to describe it. And it's rather different in many ways than the traditional analytic approaches that people learn as they're going through graduate school. So are there courses, are there summer institutes, what's a good way for people that are interested in this technology to learn how to use it and how to work with it?
Yeah, that's an excellent question. I get asked that question a lot. Unfortunately, I don't have a good answer for it. Machine learning is one of those things, one of those areas that's it's really hard to gain a working knowledge of. It takes a lot of effort. Fortunately, there's a lot online now. There are a lot of courses, online courses in machine learning. That's a good place to start. I think one of the reasons we're putting a lot of effort into these user-friendly machine learning tools is I firmly believe that one way to learn machine learning is just to do it, and for it to be easy to do machine learning, and so we're developing these kinds of user-friendly tools. Others are as well. There are open source, free machine learning packages out there. One I highly recommend is Orange. If you Google Orange and data mining, you'll find the Orange package, which comes with a very easy to use visual interface, and you just drag and drop machine learning icons and wire them up, and you can do a very sophisticated machine learning analysis using Orange. Basically build the kind of pipelines that Teapot is building, but do it by hand. So that's a good starting point, and I think trying machine learning first and getting a feel for it before you dive into the mathematics and the theory is a good place to start.
Sorry, you illustrated Teapot and machine learning in an analysis of data from a case control study. So basic etiology.
Right.
And clearly, this kind of tool, this kind of approach could be very useful in etiologic investigations where you're trying to identify possible risk factors for a health outcome. What other kinds of applications do you see or have you worked with in terms of using these tools outside of that sort of case control analysis?
Well, let me answer that in this way. Teapot can be used for any data analysis question of any type because it has access to this scikit-learn machine learning library. So it can be applied to discrete outcomes like case control data. It could be applied to continuous outcomes. For example, if you wanted to predict cholesterol levels, you could do that with Teapot. And you can do unsupervised analysis. So if you, let's say you've collected some gene expression data, some proteomics data, some metabolomics data in your population-based study or clinical sample or experimental study with a model organism, you could use Teapot to do an unsupervised cluster analysis, and Teapot would automatically pick the right clustering method to use on your data. So Teapot is very flexible because it has access to an underlying flexible set of tools. So it's really just about how you format the data, and then we have the ability in Teapot to turn on and off different machine learning methods, so you can select those functions for regression or classification or unsupervised clustering depending on what your problem is.
Thank you. A lot of machine learning tools depend on large samples, large sample sizes. Sometimes that's possible, sometimes it's not. Your case control study was not huge.
Right.
But in many settings, we're looking at that. So based on the example that you showed, it looks like Teapot at least can work fine on what I would consider to be a standard-sized epi study.
Absolutely, and I think the key to sample size is how good is your endpoint, how noisy is your data, how accurately are your biomarkers measured, how accurately is your outcome measured, and the cleaner your data is, the stronger the signal, the smaller the sample size you can get away with for machine learning. So that's how I think about it.
All right. Thank you very much. I think we're just about out of time in terms of questions. We've gotten a number of them online that we've not been able to ask. So what we'd like to do is send those to you and let you post answers on our website so that they'll be available to the folks who were watching the webinar, but we didn't have a chance to get to their question. I want to thank you for your time today, and Jonathan has some closing comments.
Thank you, Dr. Moore, for all the useful information, and thank you to everyone who participated in today's webinar. On the Mind the Gap website, prevention.nih.gov/mindthegap, you will find several resources for this talk, including the slides, references, and a link to complete an evaluation. Your feedback is very important to us as we plan future sessions. Thank you very much.